Organ transplant recipients take lifelong immunosuppressive drugs to prevent their bodies from mounting an immune response against the donated organ, yet a substantial number of them still reject the organs. A new study by researchers from the University of Chicago shows that transplant recipients also mount an immune response against commensal bacteria in the organ graft, adding to the immune response against the genetic makeup of the tissue and reducing the effectiveness of immunosuppressive drugs. The study, published today in the Journal of Clinical Investigation, also shows that this antimicrobial immune response can be triggered by immune cell memory of previous encounters with bacteria, further complicating the body's ability to accept a life-saving new organ. Before, we thought the reason why transplanted organs in humans are less easily accepted than in sheltered laboratory animals is that humans can have immune memory responses that cross-react on the cells of the organ, and memory responses are more difficult to suppress with drugs than naive responds. Now, we see that it's not only memory cells that recognize the organ itself that are the problem, but also memory responses that recognize bacteria in the organ. Two separate immune responses. The success of organ transplants depends on the type of organ. Lungs and small intestines are notoriously difficult to transplant and have shorter survival times. Statistics show that within 5 years of surgery, 41% of lung and 54% of intestinal transplant recipients rejected their grafts, compared to organs like kidneys, just 27% rejection, and hearts, 23%. One hypothesis was that lungs and intestines, but not kidneys and hearts, are exposed to microbes from the air and digestive system, and that the organ recipients were mounting immune responses not only to the organs but also to the microbes in those organs. In a previous study, Alegre and her team had shown that when mice received a skin graft colonized with Staphylococcus epidermidis, S.epi, a common bacteria found on the human skin, S.epi caused low-grade inflammation in the graft. The team then wondered if the host mounted a separate immune response against the bacteria in the graft in addition to the more well-understood, alloresponse, or reaction to the foreign cells in the tissue, and if both could damage the graft. We thought that maybe these two separate immune responses, host versus transplant and host versus bacteria, could work additively or synergistically to mount a more robust immune response against the graft and explain why the half-life of the organs that have microbes is shorter. Dealing with AL. When these mice later received a skin graft colonized with similar bacteria, the immune response was much stronger and significantly damaged the new tissue. This is significant because transplant patients already have a lifetime of exposure to many bacteria and other microbes through everyday cuts, scrapes, infections, and diet. Most importantly, when they transplanted mice with skin grafts that were genetically different and colonized with bacteria, simulating the scenario like most human organ transplants, they saw that immunosuppressive drugs that prolonged transplant survival in naive mice did not work in mice with an. That explains why when you transplant a lung or intestine, patients do less well and have to receive higher levels of immunosuppression than when you transplant sterile organs, Alegre said. You have to deal not only with the response against the graft, but also the response against the bacteria that come with the graft. Materials provided by University of Chicago. Original written by Matt Wood. Note: content may be edited for style and length. Isabella D. Parazola, Martin Sepulveda, Lu-Chu Chen, Ying Wang, Yuk-Man Lei, Jipeng Li, Rina Li, Hussein Sattar, Betty R. Theriault, Yasmin Belkade, Anita S. Chong, Maria-Luisa Alegre. Host versus commensal immune responses participate in the rejection of colonized solid organ transplants. Journal of Clinical Investigation. 2022. doi. 10. 1172. JCI 153403 University of Chicago. Bacteria in donor organs complicate immune response after transplantation. Science Daily. Science Daily, 14 July 2022. ift.tt/K1ZRTZ. University of Chicago. 2022, July 14. Bacteria in donor organs complicate immune response after transplantation. Science Daily. Retrieved July 14, 2022 from ift.tt/K1ZRTZ University of Chicago. Bacteria in donor organs complicate immune response after transplantation. Science Daily. ift.tt/K1ZRTZ. Access July 14, 2022. Thanks for watching. Please subscribe my channel for more videos.